Good day, ladies and gentlemen, and thank you for joining us for the Immutep webcast to discuss the company's new clinical data released at the European Society of Medical Oncology 2023 meeting. My name is Paul Sanger, and I will be your moderator today. The format today will be a presentation delivered by Immutep, and at the end, we will open the call for analyst questions. I will also relay any other questions received via email prior to this call. I would now like to hand over the call to Marc, Immutep's CEO, to start the presentation. Marc, welcome, and over to you.
Thank you, Paul, and welcome everyone. And we appreciate that you are taking the time to listen to our webcast. With me from the Immutep management team are Frédéric Triebel, our Chief Scientific Officer, Florian Vogel, our Chief Medical Officer, and Christian Mueller, our Senior Vice President of Strategic Development. And Florian and Christian are actually joining from Europe, where it's relatively late. Today, it's our pleasure to walk you through the exciting new results from our large phase 2 trial focused on first-line treatment of metastatic non-small cell lung cancer, which actually exceeded our own optimistic expectations. The unique chemotherapy-free op combination of our lead clinical candidate in oncology called eftilagimod alpha, or EFTI, with an anti-PD-1 therapy called Keytruda or pembrolizumab, was shared via an oral presentation by Dr. Carniceri of the Catalan Institute of Oncology at ESMO on Saturday. We will also briefly walk through the data from the phase 1 trial evaluating EFTI combined with pembro and doublet chemo in first-line non-small cell lung cancer. However, we will be limited in what we can say, given that there will be new data from this promising INSIGHT 003 study presented tomorrow at ESMO in Madrid. So please stay tuned for that. Here's our usual forward-looking statement. And before we dive into the clinical data, I would like to take a moment to discuss our lead candidate, EFTI, along with the LAG-3 environment and our different approaches and the unique position that Immutep finds itself in. As a pure play LAG-3 company, we are very pleased to see the increasing attention paid to this unique gene and also the interaction between MHC class II and LAG-3. Our pipeline is very deep and very intense. We have 2 assets in autoimmune diseases. You see at the bottom, IMP761 is the world's first agonist to LAG-3, positioned in autoimmune diseases, and we are eager to move forward towards clinical development. GSK781 and LAG-525, or iramilimab, are 2 assets we have out-licensed to GlaxoSmithKline and Novartis. We have a very early stage anti-LAG-3 small molecule program, and focus of the webcast today is, of course, eftilagimod alpha, which we have in different clinical trials and different combinations, partly together with the industry and partly without. The next slide provides specific details on the multiple first-in-class therapeutic approaches we have designed around LAG-3 and its interaction with MHC class II. I think it's quite important, and I would like to focus on one main aspect here. It's quite important to note that eftilagimod and our way of targeting MHC class II is absolutely unique, and it should not be mistaken for anti-LAG-3, which you see on the top left-hand side orange box, the classical concept of an immune checkpoint inhibitor concept we license out to Novartis, block the checkpoint on the T cell, keep the T cell active. What we do with eftilagimod is unique and activates the immune system broader and we believe deeper than some other concepts in cancer immunotherapy. So you see after activation of the dendritic cells, for instance, monocytes, you see more CD8, more CD4, more NK cells. You see a kickoff of the interferon gamma cascade. So what we do here is absolutely unique. Turning to slide 6. The powerful anti-cancer immune response that EFTI drives is shown here across multiple clinical trials and different indications, either as a monotherapy in renal cell cancer, as you see on the far left, in combination with chemotherapy, for instance, metastatic breast cancer, or in combination with an anti-PD-1 therapy in non-small cell lung cancer. The one constant among these three trials where you see significant increases in cytotoxic CD8 T cells or other anti-tumor cells, as well as important chemokines and cytokines like interferon gamma, enhancing anti-cancer immune response, is indeed FD. And more important than our belief in FD is the compelling clinical data in terms of overall survival and other key efficacy metrics that was just presented at ESMO. And with that, I will turn over the call to Christian Mueller, our Senior Vice President of Strategic Development.
Thank you, Marc, and thank you all for joining on this important call today to discuss this new exciting results from Part A of TACTI-02, also called Keynote 798 trial, for patients with metastatic first-line non-small cell lung cancer that were just presented here at ESMO in Madrid. And for all those who were not present when Dr. Castroveini presented, the session was completely crowded, and it's really hard time to find a space even in the overflow areas. So it attracted a lot of interest by the audience. I will provide an overview here today on these very strong clinical results, along with perspective on our registration strategy in this important indication in the next few minutes. And after that, I will hand back to Marc. And with that, let's just jump right into the data. Slide 8 provides an overview of the study design, most of you guys know, for which we have a clinical supply and collaboration agreement with Merck, also known as MSD outside the US. This phase 2 trial evaluating the combination of FD with MSD's anti-PD-1 pembro in a multi-center, multinational study across Europe, Australia, and the United States. And Part A of that study is focused on first-line therapy for metastatic non-small cell lung cancer patients. And one of the very few trials without chemotherapy, recruiting patients regardless of their PD-L1 expression levels. It's worth noting that TACTI-2 was established as a Simon's two-stage design with just 17 patients initially, and then there were pre-established response levels that needed to be met before expanding to 36, and then to the 114 patients we present here today. The trial exceeded all those levels, those thresholds, and a total of 114 patients have been enrolled. And this sizable patient population, we believe, makes the Kaplan-Meier curve stronger and, more importantly, also statistically more robust, as it narrows confidence intervals and providing assurance on the statistical strengths of the encouraging results we will discuss shortly. One more thing I would really like to mention, sorry, Marc, one slide back. One more thing I would really like to mention before moving on is that the PD-1 expression levels have a large impact on the clinical outcome of anti-PD-1 therapy. In short, the higher the PD-L1 expression, the better anti-PD-1 therapy works in patients. Conversely, the lower the expression, the less effective or even ineffective anti-PD-1 therapy is. I think it's important to keep in mind as a take-home message here from this slide that TACTI-02 had only 22% of patients with a high PD-L1 expression of above 50%, and 42 and 36 had a TPS score of 1 to 49 or less than 1% respectively. So no enrichment for high PD-L1 patients. And now turning to slide 9. Here we present the encouraging efficacy results in the entire patient population, also called intent to treat of Part A. So from all 114 patients. The strong response rate of 40% and high median duration of response of 21.6 months helped to generate an impressive median overall survival of 20.2 months in this intent to treat population after a median follow-up of over 2 years. And we should always keep in mind that this is without chemotherapy. Additionally, we saw an excellent 12 months progression-free survival rate of 37.7% and a 3-year survival rate of 35.8%. As detailed, the intent to treat population includes roughly 75% of the patients with PD-L1 expressions below 50%, meaning it is not expected that the anti-PD-1 therapy alone would work. These results are very encouraging for chemo-free immune oncology combination in an all-comer PD-L1 patient population. Frankly, we believe this overall survival data is superior to many chemo-immune checkpoint combinations in the same patient groups, for instance, as it was reported here at ESMO from the PERLA study with GSK's and MSD's anti-PD-1 plus chemotherapy in a comparable patient population. So really excited by this intent to treat population. Moving to slide 10, we see what we believe to be the strongest survival data from any dual immuno-oncology combination in a sizable phase 2 like this. Unlike so many IO combinations that are limited to high PD-L1 expressors, the benefit from FD extends across the entire PD-L1 spectrum, including low and negative PD-L1 expression patients. On the overall survival chart on the right, you can see the exceptional durability and high quality of the responses exhibited through overall survival across all PD-L1 subgroups, so less than 1%, 1 to 49%, and obviously also above equal to 50%. The table on the left provides a detailed overview of the superior data in terms of response rates, progression-free survival, durability of responses, and overall survival, which truly differentiate FD from most other IO candidates being tested together with anti-PD-1 therapy. So let's discuss the 3 TPS subgroups separately for a moment. In TPS less than 1%, where pembo is not expected to work at all, we have observed an overall response rate of 31%, translating into a median overall survival of 15.5 months, which matches almost the 16.4, which are reported for TPS positive patients for pembo alone. In TPS 1 to 49, the ORR was 45%, leading to a median overall survival of 23.4 months. And then in TPS above equal to 50, the ORR of 55% led to a median overall survival, which is even not yet reached and is expected to be well beyond 35 months and unprecedented by that. Overall, we are more than happy to see the initial hypothesis we had that FD could overcome, if you want to call it primary resistance to pembo, pretty well in all 3 TPS subgroups, as it's extended the number of responders and then translated it into overall survival. So for us, the study is a great success, which we would like to turn into a BLA and MLA very soon. And for this, let's have a look how it tracks if you combine TPS 1 to 49 and TPS above equal to 50. Next slide, please. In this population, we report a median overall survival of 35.5 months, which is unprecedented, together with a strong median PFS of 11.2 months and an ORR of 48.3%, together with an excellent median duration of 24.2 months. The significant 35.5 months median overall survival stands in a class by itself and will provide some more context on that shortly. As shown in 2 charts, the 12 months PFS rate and a 3-year overall survival rate are 46.8 and 45.6% respectively, and are also very promising and highlight the quality and the extended duration of these responses. As this basically means you lift the tail of the curve, it could be a key USP for this combination, especially compared to chemo-containing regimen. And also to that, we will put it into perspective in a second. Of note, we present here the data for central TPS measurement, but the data for central plus local TPS testing provides similar outcome. Additionally, as you can see here, unlike many other IO combinations that only target high PD-L1 expressing patients, and as their results in the low expressing TPS 1 to 49 groups are very weak. This is not the case with FD, as is evident with the strong efficacy data in these low expressors, which contributed significantly to the overall results here, the TPS larger or equal to 1%. So here you can see a very powerful set of clinical data around patients that express any PD-L1, who make up approximately 70% of non-small cell lung cancer patient populations. And obviously, as we have fast track designation for exactly that patient population, this represents a key area for FD's future development. Let's move to the next slide and try to see how benchmarking in different concepts look like. And first of all, we would like to start with benchmarking against benchmarking of FD plus pembo against historical results reported from pembo monotherapy in a comparable subset. I would call it's basically a proof of concept by historical comparison. And as the charts show, FD plus pembo is leading to superior response rates, PFS, and overall survival across all PD-L1 levels. So 1 to 49, above equal 50, and then obviously also above equal to 1. And this combination is doing it in a relevant fashion, as the efficacy in each of the parameters increases roughly in a range of 1.5 to 2 compared to historical pembo data. What is key, and again a differentiator for FD as compared to other clinical assets being combined with anti-PD-1 therapy, including anti-TIGIT, is that the data clearly shows that our results are not only clearly better in high PD-L1 expressing patients. We have often talked about driving superior results through the combination of FD with anti-PD-1 therapies. We have also discussed how we believe we can expand the patient population that can be addressed with an anti-PD-1 therapy. And here's an excellent opportunity to highlight that point, as the 1 to 49 make up 35% of the non-small cell lung cancer population. And it's quite a substantial part of that. Let's move to the next slide and to see how it benchmarks against standard of care, usually chemo-containing regimen. So to quickly explain the bar, so on the left in the blue, you have pembrolizumab, what you have seen on the other slide. In green, you have FD plus pembro, and then on the right, in each of the figures, you have different chemo-containing regimens with their respective data points for the respective endpoints. To provide even further evidence here on how powerful the combination of FD plus anti-PD-1 therapy can be, this slide here will provide the according benchmarking. And it centers, as you would expect, around PD-L1 expression above or equal to 1%, as this is the focus where we would like to develop FD plus pembro further and where we have fast track designation. As is clear from the chart on the right, the addition of chemotherapy has a negative effect on the duration of response for all therapies shown here. You can see it drops from around about 24 months to something like 13. This has been key for us why the prospect of a chemo-free regimen is so enticing and holds so much promise to positively impact patient outcomes in first-line non-small cell lung cancer. And we are glad to be able to share here with you today that without the use of chemotherapy, the response rates, together with the excellent durability of the responses, culminated in substantially better overall survival for these patients compared to historical controls. In the end, as you can see in this comparison, the 35.5 months are unprecedented in this patient population. And one last benchmarking slide on the next one, which we feel is also important and where we believe is a key difference for FD plus pembro compared to chemo plus a PD-1. We looked into the 3-year overall survival rates and the 12 months PFS rates, so long-term progression-free survival and long-term overall survival. And especially the 3-year overall survival chart shows the 45.6 OS rate at 3 years is well above pembro mono and also all chemo-containing regimens, and offer initial evidence that the combination of FD with anti-PD-1 therapy may be uniquely positioned to lift the tail end of the survival curves and may lead to more long-term responders and people, for more patients being alive at 3 years, which is medically quite a high unmet need in that population. And I promise too much, there's one more benchmarking slide. The main take-home message from that slide is that FD is doing all of that, what we have seen in terms of overall survival, with an excellent safety profile. The treatment adverse events leading to early discontinuations are exactly the same for pembrolizumab plus FD as for pembrolizumab alone, so roughly 10%. And this is lower than all other chemo-containing regimens, and it's just one example to explain the excellent safety profile of eftilagimod. This was also explicitly mentioned by the discussant here at ESMO. Now, we would like to briefly cover the INSIGHT-003 phase 1 trial. Slide 17 provides an overview of this investigator-initiated phase 1 trial design that is testing the first triple combination approach, utilizing FD in combination with carboplatin, pemetrexed, and anti-PD-1 in metastatic non-squamous first-line non-small cell lung cancer. The study is focused predominantly on first-line non-squamous non-small cell lung cancer with a TPS score of less than 50%, where the use of chemo makes more sense, and is assessing safety, tolerability, and initial efficacy. As reported in the abstract at ESMO last week, which had a cutoff of April 2023, 21 patients have been enrolled and treated, and a strong 67% overall response rate and 91% disease control rate were detailed in the ESMO abstract, which is also noted that patients with negative or low PD-L1 status, meaning with a TPS score of less than 50%, showed promising efficacy signals, and that the triple combination appears to be feasible and safe. These results give us continued confidence in the flexibility of FD to be safely combined in various novel formats and enhance various therapeutic approaches for multiple solid tumors. And importantly, Insight 003 may also help to further inform the late-stage trial design options for FD in the first-line non-small cell lung cancer, especially for patients with a TPS of less than 50. But we will come to that in a second. There will be new data presented tomorrow during the day here at ESMO of this promising trial. So please stay tuned, as we cannot share it here, unfortunately, today. The last comment on that study is that the study is still continuing recruiting, and we're looking forward to have the fully 50 patients recruited soon. So let's move to the next step regarding our registrational strategy and how we see that, given the strong data we have just been witnessed here together. This is a high-level overview of non-small cell lung cancer market, which unfortunately impacts roughly 1.9 million people annually and is the most frequent cause of cancer death. Well-tolerated treatment options that synergize with available standard of cares and improve outcomes across PD-L1 level are necessary in the frontline non-small cell lung cancer setting. So let's have a look into the unmet need a bit deeper. Turning to slide 20. This slide shows how FD is uniquely positioned to address all unmet needs with different concepts as presented before. As we have detailed throughout today's webcast, PD-1 expression levels have a substantial impact on the clinical outcome for anti-PD-1 therapies, and the strength of clinical data presented not only at ESMO but at SITC and ASCO last year shows FD has a significant potential to address all PD-L1 levels. This is important as many dual IO combinations that do not rely on chemotherapy have shifted to high PD-L1 expressors due to suboptimal results in all the other patients. This means they are focused solely on roughly about 25% to 30% of the market. And this is not the case with FD, as we have multiple options for all different TPS subgroups. Based on Insight 003, we believe that FD has the potential, as shown in strategy B on the right side of the slide, to create a new standard for first-line non-small cell lung cancer patients with a TPS score of less than 50%. These patients represent roughly 70% of the patient population, and particularly for patients with PD-L1 negative, there's a high unmet medical need for better therapies. But our current focus, as we have the largest data set and the most mature data set, including overall survival here, is creating an efficacious chemo-free regimen in first-line non-small cell lung cancer patients with a TPS score larger or equal to 1%. And this is where the company will move forward in terms of registrational setting, as I will explain on the next slide. This part, the population represents about 65%, and the approach is named here in strategy A. FD would be uniquely positioned, as I mentioned before, usually other approaches, just like anti-TIGIT, for instance, they target TPS above equal to 50. So how will the phase 3 look like? Let's move to slide number 21. Immutep is preparing to conduct a phase 3 study and has multiple options, as we have presented here, and given the strengths of the new data presented at ESMO 2023. Before we get into the details, let us start with the principles for the trial design as we see them. So the aim obviously would be to capture US and Europe, so EMA, FDA, as this would be 70 to 80% of the global non-small cell lung cancer market. And it will also mean usually many other authorities follow these two. The trial design, the timelines are subject to regulatory authority interactions, competent authority approval, and also other stakeholder feedback, as well as partnering discussions. So what are the practical implications? The primary objective is relatively clear. It will be overall survival, and we will have a robust statistical power of 90% and an alpha of 5%. The choice of the control arm will influence sample size, feasibility, and commercial aspects. And the comparator arm should be an NCCN1 category or ESMO recommended. Lastly, the patient population. So if we go for TPS larger equal to 1, or we limit ourselves to certain parts of that indication, also has an impact on the study design. This will all be based, as said, on acceptance by competent authorities in the key global markets, including US and Europe. And the design, for sure, should have a high likelihood of success based on the data we just have here presented. On the right side, you can see an example depicted where FD plus anti-PD-1 would be tested against the CheckMate 9LA protocol, meaning 2 cycles of chemo plus IPI plus Nivo, in a 2-to-1 randomized setting with a total of 630 patients. And the study will also have a futility built in for more favorable risk investment approach. And with these details, I would like to release you from the data heavy part of that webcast and hand back to Marc. And I would like to thank you already for your attention.
Yeah, thank you, Christian, for that detailed overview and leaving me with the more simple part here. So this slide 23 offers a summarized view of the data covered here today. And in summary, I believe we have all reasons to be very excited with the excellent overall survival data in first-line non-small cell lung cancer. I don't know what we should have been wishing for when we started the trial a few years ago. And it's, of course, great for the patients and very rewarding for the team to see what could be achieved when you combine FD together with an anti-PD-1 therapy. The strength of the data across all PD-L1 expression level truly differentiates FD plus anti-PD-1 from other chemo-free IO combinations and opens the door to not only stronger clinical outcomes for patients, but also for significant expansion of the number of patients that respond to anti-PD-1 therapies. Looking ahead, we have multiple development options to capture the entire non-small cell lung cancer market by PD-L1 status through chemo-free IO combinations in TPS greater or equal 1, or IO chemo combinations that target low and negative PD-L1 scores. So planning around our registrational phase 3 trial is in progress and under discussions with the regulators, and we hope that we can provide some more news on that in the coming months. The mature data from TACTI-002 and also INSIGHT-003, again, will be presented tomorrow, so in less than 24 hours, continues to build our understanding of FD's ability to boost the innate and adaptive immune system and provides important insights into how FD may ultimately meet diverse first-line non-small cell lung cancer patients' needs in both chemo and chemo-free settings. And with the final slide of this presentation, I believe 2023 has been a very active year for Immutep, and we are very encouraged with how the clinical data has matured throughout the year, culminating now in very compelling data we share today from TACTI-002, INSIGHT-003, hopefully tomorrow. And of course, this all in biotech land, very important from a financial position of strength, and thanks to the support of our shareholders and also a few new investors regarding the 80 million raise we closed mid of this year, so that we have a cash runway to early 2026 based on our current guidance. We have a number of upcoming milestones through the remainder of this year and also into 2024, like new data from INSIGHT-003, in a position to deliver there very, very soon. Completion of patient enrollment in the randomized TACTI-003 clinical trial, a phase 2b in first-line head and neck cancer, with top-line results expected to follow. We should have also updates from INSIGHT-005 and FD-SARC-NEO. FD-SARC-NEO is studied in the neoadjuvant setting, soft tissue sarcoma. We move forward towards clinical development for IMP 761. And of course, we hope for other updates as well. And I shouldn't take much more time from you. And I will pass back to Paul for questions from the analyst who kindly joined us today. So thank you so much.
Thank you, Marc, and thank you to your team members. So as Marc said, we will now open the call for analyst questions. We have quite a lot of analysts on the call today, so please try and keep your questions to just to one to start with, and if you don't mind rejoining the queue. If you do want to ask a question, please just raise your hand and I'll get to you as soon as I can. The first question to Marc and team comes from Ahu Demir from Landenberg. Ahu, over to you.
Thank you very much for the presentation. And first of all, I want to say congratulations to the whole team. It's great to see the impressive data and it hasn't disappointed, it just keeps improving, and also great to see the positive outcome of everyone's hard work. So wanted to say that first. I'll ask two questions if I could. My first question is, looking at the ESMO abstract and the data presentation, the data cutout is, there is a 6 months difference that we have noticed. And looking at the improvements on the overall survival, median overall survival data is significant, especially for the TPS equal and higher to 1 to 49, and as well as 50%. So I'm just curious if you could comment on the follow-up periods, like what is the longest and shortest period you have followed the patients? Does 6 months make such striking differences for the data set that we have from 114 patients?
Yeah, thank you, Ahu. Much appreciated. And of course, also a very important question to understand the data and how the data tracks. I will make some initial comments and then pass over to Christian. So last patient in, if I recall correctly, was in November 2021. And when you move forward and observe the patients, some of the patients are, of course, censored. And the longer follow-up time you have, the less censoring occurs. So the more mature the data is and shifts the curve based on the Kaplan-Meier formula to the right. So that also the move to the right on this curve can be longer than the difference of the data cutoff, which has been 6 months, let's say 6 months. But the move to the right can be much more than those 6 months. But Christian, you can explain it in better terms.
Yeah, thank you, Marc. And thank you, Al, for the question. I think the easiest is explained on slide number 10. If you later have a look there, you see that, oh yeah, we go there. Yeah, if you look at the blue and the yellow line, you can see that there's quite a plateau between 24 and 36 months. And this, if this is below the line or above the line, can make the difference for the median. And we have seen it in the past that certain patients having longer follow-up can lead to much more significant impact, as Marc just explained, than the 6 months. So that's the main reason. As you just look for one number, the curve itself has moved to the right, and then there's always the question when it basically hits this 50% mark, and this is obviously a little bit by can move differently than just the follow-up time increase. If that makes sense.
Yeah, it does. Thank you. That's helpful. My second question is more general. So you have provided some color on the registrational study. Could you maybe comment on the squamous versus non-squamous? And it sounds like the TPS levels that you have not finalized the decision yet. So we are curious when you would make the final decision, what will be the timeline for the commencement of the trial, and also the futility analysis, which would be crucial for the trial?
Yeah, also very important questions, obviously. So squamous versus non-squamous, we haven't seen material differences between the two. So we wouldn't necessarily split up, but go there with, if you like, a joint patient population. We have, of course, a scenario for the phase 3 clinical trial, which we have been showing. It's important to understand that this scenario is subject to further discussions. We had, of course, some with regulators and other stakeholders. The typical discussion points you have there, of course, patient population, TPS greater equal 1, which would be very nice to cover, as we would then practically double the chemo-free addressable market, or, which would also be very validly feasible, focus on 1 to 49 or greater equal 50. The comparator arm is, of course, always a discussion point. We had a press released earlier this year in May, also a discussion with the FDA, and it's pretty clear if you would like to recruit patients in the United States that you have to include as a comparator arm NCCN1 category 1 comparator. But there are, of course, a few different ones. We would like to have one which is registered in the European Union, in the United States. So these are discussions where we have a proposal, a robust one based on the statistics. But of course, we need to listen. We need to see how the precise feedback is based on the data and what is reasonable in order to capture the United States and Europe, so 70 to 80% of the market. And of course, also to be on target with the patient population and bring together, and this has been our effort from start, and we are working on this obviously since a few months, in terms of feasibility of the trial. If you look at the comparator arm of Ipi plus Nivo plus 2 cycles of chemo, in the real world, it's not used that much, but from a feasibility point of view, it would be feasible to run a clinical trial. You have to include the marketing perspective, the label, and so on and so forth. So it's a process where you need to carefully listen, adjust. We will definitely not be stubborn. The beauty is that our data allows us to move validly in so many different directions, and we have to balance then our resources and, of course, also some third-party perspectives, which are quite important for decision-making. And we will have a very busy year-end, and we will certainly update the market, how we are tracking and how it will look like till end of this year and early next year. I don't know, Christian, if I forgot any aspect there.
So nothing to add from my side.
Many thanks for your questions. We'll now move to Melissa Benson from Wilson's. Melissa, over to you.
Thank you. Morning, team. I just had one, particularly focusing in on the PD-L1 negative patients, where you saw the median of 15.5 months, with only 1/3 of those patients really responding. Can you give us a flavor, I guess, because we recognize that might be a bit skewed, the median. Can you give us a flavor for that 1/3 of patients who did respond? What was their kind of overall survival range looking like?
I don't know, Christian, Florian, one of you would like to take that question?
Yeah, I can give a general answer. What we have observed in the study is that that's the beauty of that combination. I think we tried to address in the webcast here and there, that if a patient responds, he or she responds for a very, very long time, and that means also, it is regardless of TPS. So also for the patients in TPS less than 1, the duration of response is not different than to the other subgroups. And interestingly, not shown here, but interestingly, that you can suspect that from the data being on the slides, that patients who have a stabilization of disease also can have a very substantial overall survival. So I'm trying to say here is for overall response rate, the duration of response, as you can see here also in line number 4, it's not much different in TPS less than 1. And then if you add the patients with longer duration or longer stabilization of disease, you also get some of those will have a pretty good overall survival. Otherwise, you could also not explain the nice overall survival we have, as we do not have overall response rate above 50% in 2 out of these 3 groups.
So is it correct in saying that 15.5 months is kind of undercutting it, if you like, because the non-responders are kind of skewing that down?
I think it's depends a bit. I think if the 15.5 look pretty good. And if as said, if you compare it to Pembo alone, where there's no data, but you can compare it against Pembo, for Pembo the buffer equal to 1%, those 15.5 track really well. I think for us, like if you look from a development point of view, the initial idea here is, or the initial good signal is that you see responses, which you wouldn't expect with Pembo alone. And now if you think further and you add chemo to it, you may come to a regimen where you have a response rate of 65, 70% maybe, which then would obviously exceed what is out there in the market.
Thank you, that's helpful.
Melissa, many thanks for your question. We'll now move on to Chris Redhead from Goats Partners in London. Over to you, Chris.
Yeah, hi there. Yeah, it kind of just comes down to, I mean, the lifting of the tail seems to be remarkable. That you're seeing the overall response rates at 12 and 36 months being quite similar. That's a very extraordinary result. But the question I wanted to focus on was this idea of the triple therapy and how you think that will fit in, because clearly you see when you add chemo, you get a decrease in the duration of response, and just kind of, I don't know what your thoughts are in terms of, maybe it's too early to say, but how you think that would fit into a treatment regime, because clearly you'd start off without chemo, because that would be, it's going to get the best duration of response. At what point do you switch the chemo for the patients don't respond? Just getting your thoughts.
Yeah, this is of course a very reasonable question as well. We believe, first of all, that this is what the data suggests, more robust given the number of patients, also the follow-up time for the chemo-free market, and based on Insight 003, subject to the data tomorrow, for the patients below 50 TPS, that we could capture the whole market, and that the different settings may require different approaches or solutions. So for a TPS score greater equal 1, the chemo-free solution seems to be very valid. And having said that, we, of course, as just explained a minute ago, we see also very strong results below 1. But one has to, of course, see what is the competition, what can reasonably be done. And for patients who would potentially require a chemo backbone, this could be in individual questions for patients with 1 to 49, but definitely typically for patients below 1 TPS score, we potentially have also a solution there, so that we provide different approaches for different medical problems here. I don't know if anyone from the team, Frederick, Florian, Christian, would like to add to that.
Maybe just quickly from my side, the way I think the investigators and also patients look at it. First of all, you want to have an effective therapy. And for TPS less than 1, there are some therapies out there which lead to median overall survival of more than 15.5 months. So taking the chemotherapy away would not lead to a more efficacious, or at least equally efficacious therapy. So it would probably not be good to do so. And that's why we believe for this subset, probably a chemotherapy backbone would be best. And the good thing with FDS, as Marc pointed out, we could address then the whole non-small cell lung cancer market with different approaches, and just shift the use of chemo to TPS less than 50, or even only TPS less than 1, dependent then on dependent on the data we have seen here.
Thanks for your question, Chris. Let's move on next to Benjamin Palouche from Baird. Benjamin, over to you.
Hello, thank you so much for taking the question, and congrats on the data. With the presentation yesterday at ESMO, I was just curious if there was any feedback from doctors that you'd like to highlight.
Well, that's a very good question, Benjamin. As Christian said, it was very, very crowded. I believe it's fair to say we got a lot of attention, but why not hear from the horse's mouth from Christian and Florian?
Yeah, I think we had what you usually have on these conferences, you have 2 abstracts presented, and then you have a discussant. And I think the discussant did a pretty good job in explaining where's the difference of FD compared to others. And then also pointed out that what we basically do here is we overcome primary resistance to pembrolizumab as we enlarge the number of patients who could benefit from that part of therapy. And the only question she was also impressed by the data, and the only question she had, and that's also what we get from all the PIs we talk to, let's make a phase 3.
Got it. Thank you so much.
Thank you, Benjamin.
Thanks for your question, Benjamin. And thanks for being patient, David. David Stanton from Jefferies, over to you.
Thanks, team, very much for taking my question. I guess my question is sort of a follow-up and an extension of the previous question. You know, I'd be interested in any feedback you've got from your partner, from Merck, regarding this data. Thank you.
Yeah, of course. Thank you, David. I have to be a bit general here. I believe everyone seeing and discussing the data has a very good reason to be excited and is excited. I mean, this is, if you would like to wish for data, this is almost, what you would wish for, I would say. So there, I believe, is a good level of excitement for a variety of different partners. And of course, we are honored that we worked here together with Merck, with Keytruda, Pembro being in general a powerful drug, and we have a very good relationship since years. And I believe it's fair to say that they are excited as well, but of course, they would need to speak for themselves.
Thank you and congratulations.
Thank you so much.
Okay, we've got time for just a couple more questions. Let's move to Thomas Joaquin from Bell Potter.
Thank you all for the presentation and congrats again on this data. I'd just like to focus in on the TPS group greater than 50%. I noticed in the presentation the median overall survival data for this cohort was not yet reached based on the follow-up period. But then in the abstract that was released, I think the data was around 39 months. So just wondering if you could provide some additional color there as to that cohort and the overall survival. Thanks.
Christian, would you like to take that one?
Yeah, I was offering directly to take that. We can go exactly to the same slide we just had. It's slide number 10. It shows us pretty nicely. So if you talk about median, it's always important to keep in mind the percent of events, and we have 40% of events in the TPS larger equal to 50, which means the median itself can move around. You have 60% of the patient are still censored. That's one thing, and the second is then the follow-up time of certain individuals change. And then if you look into the figure, you can see that the black line is right above the median after around about 38 point or 36 point something. That's probably 38.8 months, which means just certain follow-up has been increased. So this curve has basically been lifted. It was probably just slightly underneath the 50% mark because of different censoring at the time of the abstract. And what happens, you see the steps in the curves, and the steps get bigger over time. And that's because the patients at risk are less. And then you have, when a patient dies, the percentage basically of that number of patients at risk is bigger. So the impact is bigger. So if you increase follow-up, those steps get smaller. Also over at the end of the curve. And then you can have those kind of situations as we have observed here, that we had a median at the time of the abstract, and now we don't have a median, and it's not yet reached. I think the bottom line for that, I know everybody likes to compare numbers, is the median overall survival for that subgroup will be really, really great, unprecedented, and it may be 39 in the end, or it may be 40 something. So I think it's you know, the time will tell, but the data is unprecedented and really great in that subgroup as well.
Thanks for your answer. Cheers.
Well, thanks to everybody for all those questions. That's all we have time for. And on that note, I'll pass back to you, Marc, for final comments.
Yeah, thank you so much, and apologies for if there were any remaining questions we could not have been answering. But on behalf of the Immutep team, I would really like to thank everyone for listening today in the United States on a Sunday, very late in Europe, and Monday morning in Australia. And as a reminder, an archive of the webcast will be available shortly on our website. Thank you so much and goodbye.